Okay. Thank you all for joining this session. You can add this my WeChat number. If you need any question, you can ask me anytime. If you need any support in your project, you can ask me anytime, and I will be happy to provide any support. Before going to the explanation of CRISPR, I would like to give some pro tips. I have received 225 people registered for this webinar, but most of the people there, they I don't know, they didn't receive emails, or their contact numbers were not correct. So this is very important that you must double check your email and your contact number, that you have to join that opportunity, otherwise you will lose that opportunity. Next thing is, you might like provide the your WhatsApp number in correct format. The webinar contents, I don't want to go for this in detail. This is just like I will go over, give an overview of this CRISPR-Cas9 system, and the primary purpose of this webinar is how to use this practically. So I will just go through these things. Before going to CRISPR-Cas9 and gene modification or gene editing, one must need to know that why he need to do the deletion, to alter the gene, to delete the gene, to overexpress the gene, or to downregulation the gene. This is very important, that this gene can tell you about the lethality, how much it is important in the life of that organism, or function in a specific tissue or organ or a place. And this also tell us like secretion or less amount of specific proteins, how they bring morphological changes or physiological changes. This is just an overview of the application of genetic engineering. Like if we modify the gene, what we can get. This is just like the old things, and I don't want to spend time on this. History of gene editing starts with the uh, modification with the invention of the restriction enzyme in 1971. When this enzyme was introduced, so the genetist, the scientist, they could go for the to cut the specific site of that DNA fragment. So next, this there was like meganucleases, for example zinc finger nucleases, transcription activator effectors, and the last one is the CRISPR-Cas system. Other silencing sources like siRNA, miRNA, shRNAs are also available. What are talents? I don't want to talk about this. People are still working on talents, and they're publishing papers. People are still uh, interested in these papers, and they want to publish. uh, Talents are still alive, because some people, they want to use this technique, this technology, because this also has some good effects. It also has some good strategic plans, so that people can use this one instead of using the CRISPR-Cas9 technology. If someone is interested in talents, I can provide you many papers in that our WhatsApp group, and you can go through read. Zinc finger nucleases was a little modified form of talents, and this is also like the modified form of FOC1 restriction enzyme was a little modified. This was the first paper that has been published based on the zinc finger nucleases in genome editing, and this was the second, and this is still alive. Like people are still using in soybean this how to say, ZFN zinc finger nucleases are still in use. If someone is still interested in ZFN zinc finger nucleases, I will provide the papers in that our WhatsApp group, so you can read. There were some basic problems with the existing genome editing, genome engineering techniques or resources. The very first one was like one need to be very expert in the in the protein engineering, because the ZFNs and the TALENs, they are based on the structure of the protein, and if that domain is present in a different way, in a different position, so there will be more problems with that, and they can go for the off targets more efficiently. The second big problem was that it would take more time. So taking more time was also a problem, and more money, because many labs they don't have much more funds. So these techniques and these resources were not so effective. But the CRISPR-Cas9 has revolutionized the era. I think I should, yes, uh, revolutionized the era of gene editing, because uh, one don't need to be very expertise in engineering of custom targeted DNA binding proteins. This is, you just need base pairing nucleic acids, and they will go for their recognized position that are called forms. They're known as PAMs, and you will know what are PAMs in next slides. CRISPR is the new era of gene editing. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. This is a family of DNA sequences found in genomes of prokaryotes such as bacteria and archaea. There are many classes of this one. So class 2 is used as genome modification technology. This is about the CRISPR. What is Cas9? Cas9 is a protein. It is a restriction enzyme which is associated with the CRISPR species. CRISPR associated protein. There are many proteins available. For example, this is CRISPR-Cas9, and the other one is Cas12, also known as CF1, CPF1. Sorry. So is these enzymes are, they recognize these spaces, and then they attach and make a cut. So CRISPR-Cas9 combine is a sequence from the basis of a technology known as CRISPR-Cas9, which can be used for genes. Edit genes, we can remodify this word now. Edit of gene means like you can just like make changes in the gene, but now we can go for the epigenetic modification of the gene. So the most best word is to modify the genes. So CRISPR-Cas9 is a specific, efficient, versatile genome editing technology with which we can harness to modify, delete, or correct precise regions of our DNA. By our DNA, I don't mean the DNA of human beings. By our DNA, I mean the DNA of our interest. That can be plants, that can be animals, that can be fungi, or any organism. Jennifer Doudna, one of the pioneer of this CRISPR-Cas9 technology. I'm sorry, this picture is not new, so these citations might be changed now. And the other opponent to Jennifer Doudna was Feng Zhang. They were fighting for the patent, and sometime Jennifer Doudna will win that patent, and the other time that the Feng Zhang, he will win uh, that fund. So this picture also this might have more citations now. Just an overview of the uh, modification of this, like from where it started. The CRISPR very first reported in 1987. There are some clusters of repeats in the DNA, and in 2013, 2012, Jennifer Doudna published the paper, and 2013 the Feng Zhang published the paper, and this was the revolutionary period of this CRISPR-Cas9. The CRISPR system is like, as I have told you before, this is the bacterial adaptive system. When this virus attack on the bacteria, then these bacterial genome, this will goes to the spacer regions of this bacteria, and as these spacer regions are acquired by this host genome, there will be biosynthesis of CRISPR RNA. So this is the type 1. I don't want to talk about this. So type 2, this will just go, this the invaded genome will go for the spacers, and there will be biogenesis of the CRISPR RNA. This CRISPR RNA is being attached with the transactivating CRISPR RNA, which combinedly uh, become they get together, get combined, and then they go to that spacer region, and at the same time there will be a palindromic region, and this Cas9 enzyme, this will just rush to that place and will degrade that invaded genome of the virus into pieces. So then Jennifer Doudna, she modified this CRISPR RNA, and this CRISPR transactivating CRISPR RNA to genome editing. Sorry. Something went wrong. Maybe my internet is not very strong. So this is how these spacer regions were modified into gene targeted region. You can read about this in many papers. It's already available, so no need to spend more time on these things. The like the CRISPR RNA get together, CRISPR RNA will attach with the transactivating CRISPR RNA, and then this transactivating CRISPR RNA is synthetically modified to get together, and then they are called sgRNA. Mostly this is a big confusion for the new for the people who are new to this technology. They said like, what is sgRNA? So sgRNA is single guided RNA. Because this is guided by RNA, so that's why it is called guided. And single because this is the combination of the CRISPR RNA along with the transactivating CRISPR RNA, so this is single guided RNA. What is like this picture shows the mechanism of action of the Cas9. Like Cas9 will go to the find the frame region NGG, and it has two restriction domains. One is ρVC and other is H&H, and these two domains will make a break, will make a cut on each of the strand of the DNA. So the DNA will be degraded. Cas9 nuclease, how it works, and here is, you can see this strand is break and this strand is break. The modified okay, so these are the different PAM regions from the different bacteria. Streptococcus pyogenes, the very important one, and the very beginner one was from like NGG, then Streptococcus thermophilus, NGRRT. So these are different PAM regions. At which stage this system, the CRISPR-Cas9 system, is more effective during the cell division? Like, it is also important which cell type you are using, and this is also important that uh, which stage of the cell cycle is more effective. uh, NHEJ repair, non-homologous end-joining repair, mostly used for the addition of few bases, and HDR donor oligos are also like pasted. Some people, they work on the, like, they want to do they don't want to make a break on the DNA. They just want to overexpress or to down regulate their gene. So for that purpose, catalytically deactivated Cas9 was needed. So the, as I have talked about here, this one, this is row C and this H and H. These domains, restriction domains, were catalytically deactivated for this Cas9. So now this is called as catalytically deactivated Cas9 or dCas9. So dCas9 will just take your machinery to the respective site, and it will take, like, if you want to overexpress your gene of interest, or if you want to down regulate your gene of interest, that depends on the your plasmid construct, which promoters you have added for this purpose, and this Cas9 will just play a role as a cart and will just deliver your machinery. Different formats of the Cas9, so it can go for many different, you can go per repressor, you per activator, and different many other things also. uh, I will talk about this later, because one need to go for the plasmid vector, and these can be found from the 8 gene. This is this plasmid I have used personally from 8 gene. This is the uh, dCas9 domain, catalytically deactivated. This is like U6 promoter you need for the derivation of your sgRNA. This is your sgRNA scaffold, and here you need to add your promoter. What do you want to do with this? If you want to overexpress this, so you need VP64, and if you don't want, if you want to down regulate your gene, so then you need the CRAB domain. And here you need to ligate your sgRNA of your gene. How you will design your sgRNA, I'm focusing on that today. So you see this plasmid has VP64 domain, and this plasmid has CRAB domain. So CRAB domain is used for the down regulation, while VP64 is the tetramer, and it is used as a overexpression domain. In this picture you can see all these systems together and their functions. What is PAM protospacer adjacent motif? It I have talked about it. It is like 2 to 6 base pairs DNA segment. The very first identified was from Streptococcus pyogen, NGG, but later on other strains are also found that they have this PAM, and there are also some papers like they have modified this PAM, because sometimes we cannot find NGG or these other sequence regions for that site of our interest in that gene, so we cannot go for that restriction. So some scientists, they worked on it and they have modified this PAM region. Inducible disruption means sometimes you want to work with some gene that is sometimes you need to work with a certain gene that is like age dependent, that you need to work on that gene in a specific age. So like mice is old up to 6 months or 7 months, so then you have to go for that gene overexpress or down regulate. So for that you need the inducible gene disruption, and that is a little different. Protospacer adjacent motif, as I have told you before, are modified into different nucleotides. You can read this paper, you can understand types of Cas9. There are like wild Cas9, which will cause uh, double strand breaks used for the knockout. There is dead Cas9, which will not make any break. You can do the knockdown or the knock in of their gene like CREP, Pp64, and there is nickase. Only one domain, only one restriction domain is being catalytically deactivated. So you can make only a single strand break, and that is also useful. Like off target part, that one is a little controlled. Crystal structure. Uh, Crystal structure of the Cas9. This paper you can read to go for the crystal structure of the Cas9, and I'm not going to explain this. What we can do with the help of CRISPR-Cas9 gene editing technology? This is a big question, and the answer is very simple and just few letters. Imagination is the limitation. We can do like many things with the CRISPR system. I will use the CRISPR system term. We can do many things: gene modification, single base chain, gene therapy, cancer treatment, drugs design, and nowadays the Feng Zheng, he also introduced a kit for the diagnosis of the COVID-19. And there are so many other uses of this system. Some people they have used for some unwanted unethical issues. I don't want to go for that discussion. You will think that I am just in a rush and I'm going very fast and not explaining many things. This is just because I'm not agreed to spend more time on the introduction. Uh, I I want to use more time on its practical, how you can use it in your laboratory. So please allow me for that. What's new in the world of CRISPR? The very new one is RNA editing, and this is awesome. And single base editing, it's also very useful. We have done in our lab. My friend Salah, he has submitted his paper, and we are hopeful it will publish soon. Maybe I think he's here with us. My internet is playing with me today, I don't know why. Okay, so news about CRISPR-Cas9 to treat the patient with genetic disorder. This article is really interesting. Uh, like, uh, should the rich be allowed for what? To buy the best genes? What is your answer? I would like to listen to your answer in the comment box. My answer is yes. US government is using CRISPR to create radiation immune super soldiers. You can read this article. This is very important, and this is what my webinar is based on. Ask a few questions from yourself before designing up a project, such as what do you want to do with the gene? Why you want to play with this gene? You want to knock out this gene, knock down, knock in, or you want some epigenetic modification like hypermethylation, hypomethylation, or phosphorylation, some other things. What is your target organism? This is really important. Your target organism is mice, fish, C. elegans, plants. Then which plant? Like Arabidopsis or rice or any other. Have you identified your gene of interest? You might know your gene of interest from your mentor, or you might know about your gene of interest from the literature. There is a problem. Someone might have done work on the same gene, so you have to think differently. how you should think differently. For example, someone has knocked out this gene from the whole body, and he will give you some like some explanations, but still there are so many questions unanswered. For example, that gene has more expression level in heart, in brain, in lungs, so you should go to target only that organ for that gene. And expected time, how long it will be finished, this is a long story. You just have some estimations. Usually in mice, this take up to like up to 2 years at least, if you are very efficient and if you are very expert. Gene of interest, here we are going to talk about GATA1 from GATA family, and I'm going to explain how you will find your gene of interest from NCBI, and then how you will go to tally your sequence with the ensemble, and then how you will design your sgRNA, how you will find your vector, plasmid vector, and so on. This is, you can go for the strategic plan to knock out this gene. This gene has 5 exons, so you can design your sgRNA, one sgRNA upstream of exon 1, and the second sgRNA you can go for the downstream of your exon 5. Then you might have your forward primer and your reverse primer. So by going for this during the PCR, you can know that your gene is deleted or knocked out or not. This is the plasmid. You have to go to, this is the plasmid name, and you have to digest with the BBS1. I have shown the picture before, so it will become a single strand. Then you have to gel purify, and then you have to ligate your sgRNA, and then you have to go for the sequence confirmation of that your sgRNA, either it is successfully ligated or not, and then you are ready for transfection. Gel purification, measurement of gel purified plasmids, ligation of sgRNA to linearized plasmid, transformation, maxi prep, confirmation of sgRNAs with sequencing, then microinjection in case of mice, or you have, if you are working with cells, so then you can go for the transfection using the commercially available kits, then POPs, genotype, and further experiments. Thank you. Thank you so much. And now we are going to go to the NCBI, and the gene of interest I have told you is what? GATA1. So first of all, I will go to open the NCBI website, National Center for Biotechnology Information. All databases, I don't want to go for all databases. Let me make it full screen. Yes, you can see. All right, so we need to go to NCBI. I don't need all databases. I will go to select nucleotide, or you can also select gene. You can also select gene, but I will select nucleotide. My nucleotide, my gene name is GATA1, and my organism is musculus mice. Here you get a lot of information. You don't need to go for all of these. You play with this, you will understand, and you need this one, GATA1 binding protein 1, musculus house mouse, also known as, these are the other names of also This gene have some other names also. And this is gene ID. Gene ID might be only single, only one. There can be many names but only one gene ID. I will click on this GATA1 binding protein 1 to go into depth of this gene. When it is updated for June 2020, this box gave you summary of this gene, its official name, official full name, GATA binding protein 1, provided by who? MGI. MGI is a very mouse genome informatics is very useful website, and I will show you more information about this gene on MGI. C related, I will show you this one. Gene is protein coding. If this gene is not protein coding, so then you may not think to go for its knockdown or knockout or some other things for your experiments, because if it is not coding for protein, so it may take very long time for you to get your final results. That deletion of this segment in gene can give you this results or this or this. So I will not suggest you to go for if the gene is not protein coding, means pseudogenes. Okay, so it is also some more information. Here is the explanation a little bit more, and down they showed you the expression level of this gene in different parts of the mice. So the most higher expression level is in liver. Why? You might know that GATA1 is a hematopoietic transcription factor, and liver is the base for the hematopoiesis, while the newborn bulb, later it will goes to the bone marrow. Okay, so now let's go to the find the sequence of this gene. We need to click on this gene bank. All right, this shows us the presence of this gene on chromosome, like the my strain chromosome, its position. If you click on this whole sequence, this will give you the whole sequence for this chromosome, which will take very long time to download, and you don't need that. You just need, they have already selected. You can play with this one also if you want. Show sequence, show reverse complement. Sometimes there is a problem with this display options. Make sure that your gene is present on positive strand or negative strand. This is very important. This gene is 17,404 base pair long. So we go down, and this is the sequence of this gene, started from 1 and goes up to 17,404. Now it depends on our experimental plans, what we want to do with this gene. Let me consider I want to delete this gene, I want to knock out this gene. But if you want to do overexpression of this gene, then you have to find like the ATG, the start codon of this gene, and you have to target your sgRNA to that start codon, so that Cas9, deactivated Cas9 will bring all the transcription machinery to the start codon. We have published one paper from our lab very recently, and we have downregulated VEGF. My friend, she published the paper. So this will bring all the transcription machinery to that start codon. And now if you have added your which domain, downregulation domain or overexpression domain, so that will work accordingly. I want to find the start codon, so I need to go for the CDS coding sequence. So you see coding sequence start from 13442, 13442. So let me see if this is ATG, so my gene sequence is correct, and if it is not ATG like the start codon, so my gene sequence is not correct and I have to modify my search again. What's the number? 13442. 13442. Yes, this is 13441 and this is 13442. Yes, ATG. So this is the start codon for this gene. Now what I want to delete this gene. I need my sgRNA somewhere round about this start codon. I can go upstream also, I can go downstream also, but keep in mind that for deletion of the gene, you might use two sgRNAs, like to cut the DNA fragment in two places. So this is more efficient. I want to make sure, double check the sequence of my GATA1. So I want to go to another website, another database to cross check the sequence of my GATA1 and to cross check the start codon. So I will go to Ensemble. Mhm. What's wrong with Ensemble? Yes. So Ensemble is also almost the same database as with NCBI, which provide almost all the bioinformatic information. Search species, all the species they have are available in this Dropbox menu. My organism is mouse, so I will search for it, mouse. And what's my gene name is GATA1. Then go. You got uh, many information as an NCBI. You don't need to go for all of these. You go for GATA1. You mouse gene. Let me make it a little zoom in so more brighter. All right. So GATA1 strain. This one. The rest are the transcript variants. All right. So we got the information of this gene, gene synonyms, other names also, where it is present, reverse strain. So here you see by default they are showing you reverse complement. Why? Because this gene is present on reverse and negative strain. Let's go to our primary purpose. So by clicking this you will see more information. GATA1, these are transcription variants and you need to go for the highest one. Summary of the gene and many more things. Let me click on this one. Yes, and after clicking on this one I will go here to see exons. You know, exons are the sequence, exons are the regions where they are translated regions. Okay, so our first was exon was might start with ATG and it is here. So let's see, ATGG, right, and followed by three C's. Let cross match. One C, two C, three C. Okay, and ATGGA followed by one, two, three, four T's. One, two, three, four. Yes, it means that our sequence of GATA1 is correct, and our start codon we identified at the exact position, and we have decided to knock out we have to decided to knock out this gene. We can do anything with this gene later. If you need more information about how to do that, you can just send me a message, I will explain. For making sgRNA, there are so many databases available, and today I will talk about only two databases, and then I will also tell you the most efficient one and the one I like the most. So now let's go to Centigo. Centigo engineered cells and CRISPR kits. We will go to their website. Yes, we got this website. Centigo would like to send you push. No, don't allow. Okay. So here you can read this website. There are many useful information in this website, but what I need, we will go to bioinformatics CRISPR design tool. Yes, CRISPR design tool, world's fastest and easiest CRISPR gene knockout design tool. You see, gene knockout. If I want to knock in the gene, then I cannot use this tool, this website. So that's why I will not recommend this website to use. I will just like have an introduction, launch the design tool. Type a genome name or ID. So our organism is mouse. Gene name GATA1. And now they are searching a nuclease. Cas9 is from Streptococcus pyogenes Cas9. Search. Now the system is working and they're calculating and they're working. Good. So they have searched all the databases and they gave us these four sgRNAs as they think these are the best one. You can play later with this website, and I don't want to spend more time here, so I will just close it. The best one which I like is benchling, because benchling because I can control, this is cloud-based informatics platform for life science. I can control many things. In Centigo, I cannot control many things, but in benchling, I can control many things. On benchling, first you need to go to sign up. I have already account, so I will go for sign in. Let me see if I remember my password. I will suggest you might go for the sign up with your school website. School website, I mean, that have edu. Huh? Yes, yes, yes. That that is based on edu, so that can give you some uh, more services, like you can get some um, more space in GB, something like that. Incorrect password. Password is a big problem. Hmm. Again, go back. Create an account. This email is already exist, so I will do the forgot password. We just sent you an email. Password, let me see. Okay. Sign in. So now we are here in this database. This database is very complex, and it will take about up to 1 month or 2 months to understand it completely. But you don't need to understand it very completely. You just need to know how you can design your sgRNA by using this website. So here in this bench link, you will go to this plus. Is it okay, or I should zoom it in? Let me shall zoom in. Okay, you go to this plus, create, play with this. You will know what is this. Play CRISPR, uh, create CRISPR and CRISPR guides. What is, how do you want to import your sequence? By gene name, chromosomal coordinates, sequence file you have downloaded from NCBI or from Ensemble, GenBank or FASTA file, raw basis. You have your basis in your Word file, and you just copy paste them. I will go by gene ID, search by gene ID, got one, and they will go for the rest. Genome, you can select the organism from here. These are all the organisms available on this database. So we need to go for musculus. Transcript is our GATA1, sequence is GATA1 sequence, and now go for, you can choose your folder. Where do you want uh, to save it? What do you want? Single guide, wild type Cas9 single guide RNA higher efficiency. Paired guides, you need cases, that is a little complicated. You need the gas for base editing. You want to change single bases. Our genome, your PEM, which PEM you are going to use. Oh, I got a message. There is no screen. Let me check it. My screen is here. I have shared the screen. Okay, again. It's okay now. Please give me a response on my WeChat. If you can see the screen now, so please let me know. Still no. I have checked. Can you see it now, please? Oh, so what's wrong? Any other member, please give me a reply on WeChat. Either you are seeing what to do. Okay, so I have to stop it for a while and I will log out and then I will come back. That might fix the problem. Thank you for informing me on real time. Okay, so can you see it now? Still no. Okay. Dear friends, if I log out, that may affect uh, our meeting ID. So I don't know if it is changed. So either you can. Oh yes, it's because of the time. 52 minutes gone, so the screen is disappeared. I will end, and if it is new meeting, so I don't know how you can get entered into new meeting. uh, all of your friends, you have my WeChat and my WhatsApp number. Please send me a message on my WeChat and WhatsApp, so I will give you a new ID of meeting, new meeting ID. So I'm really sorry for this. This happened to us. All of you, you have my WeChat. My WeChat is Noor, Noor, Noor underscore BAHADAR. So if you don't have my WeChat, you can add it. Otherwise, you can send me a message on WhatsApp. My WhatsApp number is 0092 345 9499 421. I will repeat. WhatsApp number is 0092 345 9499 421, or my WeChat is Noor underscore BAHADAR. Thank you.